So in the previous lecture we have studied I hope all of you can see the screen. So we are continuing with the chapter Heredity and Variation. In the previous lecture we have studied about heredity, inheritance, genetics, inherited traits, and expression of traits. Also, we have studied chromosomes and the different types of chromosomes, right? In this lecture, yes, in this lecture, we are going to study about DNA. Now, it is an important topic with respect to your exam also and with respect to the knowledge as well, right? So this topic or this you should know forever in your life also, it is going to help. So the next point is DNA. DNA, the full form of DNA is deoxyribonucleic acid. As all the information all the information is stored in the DNA, and it is inherited or it is transferred from one generation to another generation. So that is why it is called as the genetic material, right? So that is all the heredity and the variation depends on this DNA, right? So this is how the DNA looks. You must have seen the picture of DNA, right? Okay. I am sharing the screen. So this DNA, this was first time it was discovered by a scientist called as Frederick Mesher. So you can see all the information is written in front of you. It is very interesting. See, he began his work with the white blood cells in 1869, right? So he was studying white blood cells. You know, in blood there are white blood cells and RBCs and WBCs, right? So he was studying about WBCs. So while studying this in 1869, white blood cells are the major component of pus in the infections, right? As a result, Misher collected a lot of pus from bandages at a local hospital. Then he added a weak alkaline solution to the white blood cells. When he did this, the cells decomposed and caused the nuclei to move out of the solution. Right from the nuclei, Misher isolated a substance known as nuclein, and after chemical analysis, nuclein was later renamed as DNA. Right, so he was the first person to discover the DNA which is present in the nucleus. Okay, Friedrich Mischer, you can see this. Then, so this DNA is present. This DNA is present in all the organisms. It is present in bacteria, it is present in viruses, all plants, animals, human beings. So the DNA is present in all the organism, and the structure of the DNA is also same in all the organisms with some differences, right? The location of DNA. Before I show you the location of DNA, I hope everyone must be knowing it. And previously, it was thought that the DNA is present in only in the nucleus. That is why the name of this DNA is also called as nucleic acid, right? The material which is present in the nucleus, that is called as nucleic acid. But later they have discovered that the DNA is not only present in the nucleus, but it is also present in the other organelles of the cell. It is also present in ribosomes. It is present in mitochondria, right? But a lot of DNA is present in the nucleus. Still, it is called as nucleic acid. So you can see with the help of this picture, it will clear your concept how the DNA is located in the cell. You can see here, this is a cell. You can see the animal cell, then you can see a nucleus in this. So inside the nucleus we have studied cell division. Inside the nucleus there are chromosomes, and these chromosomes are clearly visible during the metaphase of the cell cycle, right? So in this chromosome the DNA is compactly, that is, it is recoiled and it is very compactly filled in this chromosome, right? So you can see here, so DNA actually is a very long structure. You can say it is 1 meter of the DNA is coiled to form a chromosome, which is so small that we cannot see with our naked eye, right? It is in a cell, so you can imagine a cell is there. Inside the cell, nucleus is there. Inside the nucleus, you are having chromosomes, and then in chromosomes, these DNA strands are packed, right? So they are very thin, but they are very long structures. So I hope you have understood the location of the DNA in the cell. Now you can see the uncoiling of the chromosomes. So this DNA is present in the form of chromosomes. So you can see how it is coiled. So chromosomes are mainly made up of the DNA and the proteins. This DNA is coiled around these histone proteins, which you can see here in the shape of balls. So this thread-like structure, DNA is coiled around this ball-like structure, that is histone protein, and in this form they are coiled and they then form a supercoiling, and the structure is formed that is called as chromosome, right? So this you can write it down. If you're having a pen in your hand, note this down in your notebooks. What is it? DNA. What is the full form of DNA? DNA is DNA is deoxyribonucleic acid. Chromosomes are mainly made up of DNA and proteins. Then the DNA, it was first discovered by a Swiss biochemist, Friedrich Mescher, in 1869 while he was studying white blood cells. Then they are present in the nucleus, so they are called as nucleic acid. But you can also mention they are also present in mitochondria as well as ribosomes. Then this DNA is present in all the organisms, right from viruses, bacteria to plants, animals, human beings, in all the organisms. And the shape of the DNA is also same in all the organisms. Then it controls, this is important, it controls the functioning, growth and division of the cell, and therefore they are called as the master molecules, right? So all the functioning of the cell, then the cell division, that is reproduction of the cell, growth of the cell, so everything is controlled by this DNA. So that is why these are called as the master molecules which are present in the cell. As our whole body is controlled by the brain, so the cell is controlled by this DNA, right? So that is why these are called as the master molecules, or in other words, you can also say DNA is a life, right? Any organism which contains DNA, or the substitute in some rare viruses, it is RNA, then those organisms are said to be living, right? They are alive. Okay. I hope everyone has written this down. Students, I can see you. Some of you are writing, the others are not writing. Write down so that you remember. Okay. Okay, so now we'll study the structure of the DNA. Okay. So in 1953, Watson and Crick, these are the name of the scientists, they have proposed a double helical model of the DNA molecule, right? So now the diagram is important. These are the you can see the faces of the scientists. So this is also important. You should be able to recognize the scientists. So these are Watson and Crick, and you can see the model of the DNA, and they were also awarded a Nobel Prize for this. Yes, all of you can draw this diagram. This is the structure of the DNA. It is called as a double helical structure of the DNA. As you can see in the diagram, this is called as the sugar phosphate backbone. You can see two parallel strands of the DNA, and here you can see the horizontal lines which you can see, or the slanting lines, those are called as the nucleotides, right? So two nitrogenous bases, they combine to form a nucleotide. So two parallel strands made up of sugar and phosphate, that is called as the sugar phosphate backbone. This is one, and this is another, and they are coiled. You can see it is a ladder shape, but it is a coiled ladder, right? So you can see here, as per this model, two parallel threads of nucleotides are coiled around each other. This arrangement is called a double helix. This structure can be compared with a coiled and flexible ladder, right? You can imagine a ladder, right? You know, a ladder which we climb to go somewhere, to go upstairs. So yeah, if you coil that ladder, so DNA structure is the same. Each strand in the molecule of a DNA is made up of many small molecules, that is known as nucleotide, right? So as you know, for example, if you take the example of a sugar, what is sugar? You must have studied in photosynthesis also. We see the sugar molecule, right? So the sugar molecule is made up of the many different glucose molecules, right? C6H12O6, isn't it? So when many glucose molecules they attach together, they form a sugar molecule. So in this way, when many nucleotides they combine, they form the DNA molecule, right? So we can draw a rough diagram. Okay, now we'll see the molecular form of DNA. So there are four types of nitrogenous bases in the DNA, that is adenine, guanine, cytosine, and thymine. Right? Adenine and guanine are called as purines, while cytosine and thymine, these nitrogenous bases are called as pyrimides. In the structure of nucleotide, a molecule of the nitrogenous base and phosphoric acid are each joined to a molecule of a sugar, right? So you can see there are three components which form the DNA. Those three components are sugar. Can you see here? This is a sugar molecule. Then here is a phosphate group. This sugar and phosphate, it combines, right, to form the backbone of the DNA molecule, and this is the nitrogenous base, right? This is adenine. This is the structure of adenine, right? So as there are four types of nitrogenous bases, nucleotides are also of four types. Now you have to remember that adenine always pairs with thymine and cytosine always pairs with guanine. It is also called as ATGC. Adenine will always combine with thymine and cytosine will always combine with guanine. So you cannot write AG or TC, you cannot combine like this. If you are making a gene or if you are writing a sequence on the DNA, it should always be AT and GC, right? So it can be AT GC or GC AT or AT GC, right? Only these four nucleotides or in only these four nitrogenous bases, they are arranged in a different manner and the whole DNA is formed, right? So only these four nitrogenous bases, they are combined in a different sequence, and the whole DNA is formed. And three group of this nitrogenous bases, for example, AT, GC, and then again AT. So this three group it forms one gene, and it is responsible for the different characteristics of an organism, right? The molecular DNA, right? Molecular DNA means now we are going in details, right? So first we have seen how externally the DNA used to look, a sugar phosphate backbone, and in middle the nitrogenous bases. So here you can see this is what is the DNA. Here on both the sides, this is sugar, this is phosphoric acid, sugar, phosphoric acid, sugar, phosphoric acid. That way the backbone is formed. Here also on the other side, the this is sugar phosphate, right? So these are the two strands of the DNA, and in between the horizontal lines of the slanting lines, which you can see, these are formed of the nitrogenous bases. So these are C. You can see here it is written A. This is T. This is G. C. C G A T. Right. So that is nothing but adenine, thymine, guanine, and cytosine. These are the four nitrogenous bases which are arranged in a different manner to form the DNA. This is nothing but the sequence of the DNA. Okay, so before we start with the gene, so I would like to show you a video so that you can clear with your concepts, right? So that video will help you to understand, right? Now, see, previously it was difficult to teach all this, but now it is very easy, and for students also it is very good that they can see live how is the cell, how is the structure of the cell, how the chromosomes look, how the DNA look, right? So now even now you can imagine also, as well as you can see with the help of video, and you can understand, you can clear your concepts, right? So I am sharing my screen, and we are going to watch a video. Watch the video carefully. Yes, I hope it is audible. Almas Miss, is it audible? The video? Okay, I hope it is audible.
मैडम आवाज बिल्कुल नहीं है। हेलो मैडम।